start your PPT.
Okay, yeah, thank you. So, good evening to all of you, my friends from China and India. So, it's my privilege to speak in this lecture. So, I'm moving to my title, that is Targeting the Driver Genes p53, EGFR, and KRAS in non-small cell lung cancer using natural products. So, as it has been discussed very previously about EGFR, KRAS, and other genes, which get mutated and they make a very difficult kind of non-small cell lung cancer. And actually, they make it metastatic making. Therefore, comes the role of different kinds of medications. So, taking on to the notes, I would like to speak on this topic very quickly. And as we can see from the global cancer statistics, there are around 9.3 million cancer cases and almost 10 million cancer-related deaths that occurred during 2020. The cancer from female breast has surpassed to the lung cancer as the topmost cancer with 11.7.3% new cases, while lung cancer remained to be the second one, very close to breast cancer, 11.4%. The critical part from lung cancer is that it is still the leading cause of cancer-related death that has accounted for around 18%, and that is somewhat around 1.8 million cancer-related deaths, mainly due to lung cancer, followed by colorectal cancer, liver cancer, stomach, and breast cancer. So, when we are seeing this picture, that is basically countries ranking towards cancer-related mortality, there are different grades, like kind of first grade, second, third, and etc. So, I can see that there is a diversity of mortality. I'm belonging from India. Our country comes to the third rate of the mortality, while rest of the world, largely Europe, is suffering from high-grade mortality. USA is suffering from high-grade mortality. So that has become a critical concern. And there comes the role of basic sciences and clinicians to work together. In terms of gender-wise cancer cases worldwide, we can see that breast cancer is top in females, while lung cancer is top in males. And collectively, they make it second most treated cancer across the world with 11.4% cases. And that's what makes it a very critical component to understand and to analyze. And there comes more specific role of one of the very important components, that is tobacco. Consumption of tobacco is more prominent in males, therefore it suppose that lung cancer and oral and squamous cell carcinoma that occurs in males is mainly due to tobacco. However, when we go to lung cancer progression and staging, there are total 6 different stages, as mentioned from A to F. At the early stage, we can see that cancer cells present in the airway mucus, but they do not form tumor. This is at the very early stage and very easy to cure, and clinical manifestations are easier. At the later stage, that is mentioned as a small tumor formation, and the tumor size is less than 3 centimeters. Until now, the patient remains quite comfortable and it is still treatable. At the later stage, the tumor grows and it becomes more than 3 centimeters in size. Now, this is the time when it starts to block bronchus, the bronchioles, and other respiratory trachea, and it causes respiratory discomfort in patients. And the next step is spreading of the tumor from lungs bronchus to the rest of the body. And the later stage, it can even cross through different membranes and can go to heart, liver, brain, bones, kidney, etc. That is the commonly called as the fourth stage or the metastatic stage. So it takes a very rapid succession, and from one patient to another patient, it can be somewhat like from few months to few years. In a very nutshell, the lung cancer can be defined as the early stage with smaller tumors up to 3 centimeter and the late stage with more than 3 centimeter and metastatic tumors. And they have high progressive rate and usually untreatable in nature. This is a very similar picture from the previous one. Here we have added two major components. One is genomic factor, the environmental factor. Environmental factors, they mainly are associated with smoking, air pollution, high risk occupational hazards, age-related disorders, and other lung diseases. The genetic factors, there are up to two types, the genetic and epigenetic factors. The genetic factors mainly include different types of mutations that we were listening in the earlier generations. And here comes the role of NGS, next generation sequencing, and other technological developments to identify these mutations. So your efforts in identifying these mutations and then targeted therapy comes from this particular point. And that is why it is very important to be considered. The epigenetic events are associated with DNA methylation, histone modifications, chromatin organization, etc. Altogether, the alterations and aberrations ultimately lead to tumor metastasis. The second important component in lung cancer is understanding the tumor microenvironment. Every normal tissue, either lung or any other tissue, it has particular immune cell system or immune microenvironment. They contain certain macrophages, neutrophils, dendritic cells, lymphocytes, CD8, and different types of cells. What happens when carcinogenic process starts? There is a sudden change in immune profile, tumor microenvironment actually. At the early stage, CD8 T cells, dendritic cells are exaggerated. And at the later stage, different MDSCs, M1 and 2s, neutrophils, and regulatory T cells, they are flooded. And they make a cytokine storm in the lung fibroblast. This ultimately leads to tumor generation and invasion of tumor from one side to the other side. From a staging point of view, at the early stage of lung fibroblast, the activation of TGF-beta makes it to cancer-associated fibroblast. And this is the preliminary stage when the cell starts to change its morphology. As we can see in this picture, the cell morphology is being changed, and it's somewhat like changing from the typical phenotypic structure to the different phenotypic structure. Then in the last stage, epithelial to mesenchymal transition starts to occur, EMT. And this is exaggerated with the help of different inflammatory cytokines and chemokines, especially IL-1 beta, IL-6, and TNF alpha. This makes them migratory cancer and mesenchymal type of lung cancer or the lung fibroblast, which later starts to induce angiogenesis, vasculogenesis with the help of VEGF, and abnormal blood vessels start to form. Later it becomes a metastatic tumor and it causes immune invasion. Different types of immune cells start to flood from one side to the other side, and they make it a complicated bronchial disease with metastatic tumor. There are some regulatory factors that we commonly call them as driver genes. One of the most potent genes is p53 because this is one of the vital signaling component which is associated with so many pathways, starting from cell proliferation, regulating the different genes' transcriptional activity, genomic instability, autophagy, apoptosis, and so many metabolic processes that happen within the cell. And one of them is DNA damage and repair system. Once p53 is mutated, at different level of mutations at different amino acids, it causes different structural changes and functional changes. And therefore, it exacerbates the carcinogenic process that happens within the lung fibroblast. Now, when you take a note from p53 dependent pathways, it alters several signaling pathways in glucose metabolism, fatty acid metabolism, amino acids metabolism, and it affects mitochondria and leads to reactive oxygen species free radicals or the oxidative stress. When we define them, there are two major components where p53 directly interferes. One of them is cell survival program, the other one is cell death program. In case of cell survival program, it helps in maintaining oxidative stress of the cells. They raise antioxidants. However, when p53 is mutated, this helps in DNA damage repair. It helps in maintaining cell cycle. But arresons, alternations, mutations in p53, they indirectly cause aberrant apoptosis or lower apoptosis, aberrant cell cycle, and skipping of the stages of cell cycle. So this is a very critical gene that can be targeted in cancer, including lung cancer, for different type of target agents. And there comes the role of different natural products. In the previous lecture, we heard about different type of compounds and the therapeutic regimen that can help in controlling lung cancer. And we know that being from South East India, China, South Korea, Japan, these countries, especially traditional Chinese medicine and Indian traditional medicine, they are very rich in those plants and other substances who can help in maintaining the lung cancer, or not only lung cancer, many other cancers. They have different subsets starting from polyphenols, terpenoids, etc., who can manage from the development of normal tissue to abnormal tissue, and they can work at different levels. For example, some of the critical factors are they can regulate cell viability and proliferation, they can induce cellular apoptosis process, they can induce lipid peroxidation, they can reduce nitric oxide levels, thereby they can suppress cell proliferation and they can suppress oxidative stress. Therefore, they can reduce lung cancer development and progression. There are so many ways these compounds and these natural products can be helpful in our anti-cancer drug development program. For example, taking notes from some of the popular plants like orphanum,